Hi there, and welcome to Next Week Today. I'm Hannah Watkins, a PhD student from Simon Fraser University, and I'm coming to you from this blank void that's been my home office the entire pandemic. It's been a busy conference so far, with presentations spanning topics as varied as charismatic megafauna to charismatic microfauna, but for the next 12 minutes, we're going to dive into the slightly more unconventional. Our main story today is the sea cucumber, an animal you might recognize from some oddly dramatic Nat Geo and BBC videos.
These animals may look like boring lumps, but sea cucumbers have all sorts of surprises. It knows no fear, possibly because it has no brain.
Okay, it's revolting, but could this walking colon be a safe haven?
And while we haven't even gotten into evisceration, rectal-dwelling pearlfish, or anal teeth, sea cucumbers are a lot more than just a fun blue planet feature. In Asia, particularly in Hong Kong and mainland China, sea cucumbers are actually incredibly valuable as food and as medicine, and can be worth as much as $3,000 a kilogram. The problem is, this incredibly high value drives a huge demand for more product, making sea cucumbers prone to overexploitation. Since they're so easy to find and collect while diving, harvesters can easily fish out an entire area and then move on to the next. And this is a pattern we've seen time and time again over the past 50-some years. Areas close to the primary market for cukes get cleaned out, and new fisheries open further and further away. Perhaps unsurprisingly, this puts cucumber populations in a bit of a pickle. But it's possible that these fishing practices can hurt more than just the cukes. Most of the pop science articles and videos on cucumbers, like some of the ones we saw before, suggest that they're the key to solving everything, from algal blooms to ocean acidification, and that if we fish them out, it'll have some pretty devastating impacts on their populations. This is based on the idea that as cukes mosey around the ocean floor eating detritus in the sand, they release key nutrients that plants and algae need to grow, they reduce the organic matter buildup in the sediment, and they break down calcium carbonate that corals need to grow. And while I'm all for assuming that sea cucumbers are more important than fish and other animals in marine ecosystems, because frankly, fish get all the attention in marine biology, and I'm really sick of people telling me that these weirdos are so important, if we dig a little deeper into the sweeping generalizations about sea cucumbers, we can see that we don't have all that much evidence to back them up. For instance, a 2010 paper from a team in Australia is often cited as proof that seagrass meadows are screwed without cukes. But the paper itself presents rather modest results. Across 2 years of cage experiments with 4 different response variables, the team found only a single significant result. In 1 year, the cages with no cukes, shown on the right here, had slightly lower growth rates than either of the control treatments. Another popular study, containing what is without a doubt the greatest figure ever published in a peer-reviewed journal, projected the total services provided by a population of sea cucumbers through bioturbation to a large reef in Australia over a year. And while this can yield really valuable insight into the magnitude of services cucumbers can provide, the authors never did test the impact or lack thereof of this service. And so, even with all this really cool research going on, we still don't have a clear understanding of how sea cucumber fishing really impacts exploited ecosystems. So we thought it would be worth re-examining one of these services, specifically testing the hypothesis that primary productivity in nutrient-poor environments is driven in part by nutrient supply from sea cucumbers. And we conducted our work in Rock Sound out of the Cape Eleuthera Institute on the island of Eleuthera in the Bahamas. Now, fishing in the Bahamas is an incredibly important industry, but sea cucumber fishing, not so much. Apart from in North Andros, in which a small export fishery was opened in 2010 before being closed within the year due to massive stock declines, sea cucumber populations throughout the rest of the Bahamas, including those in Rock Sound, remain unexploited. Rock Sound is also home to a huge network of patch reefs and seagrass halos, which make these fantastic natural replicates, meaning we can scale up from small-scale cage experiments to something more reflective of an entire ecosystem. And there were really 3 things we wanted to find out with this project. What was happening in the system before anything happened to the cukes? What happens to the patches overall when we do meddle with cuke densities? And what happens to individual plants? And to answer our first question, we visited 21 patch reefs with varying sea cucumber densities and collected data on 5 metrics of seagrass: above and below ground biomass, blade heights, shoot counts, and elemental composition, specifically looking at the percent nitrogen composition in the blades. To answer our second question, we then manipulated sea cucumber densities across these patches to 3 different levels: control, half removal, and full removal, representing 3 different fishing scenarios. Over the following 3 months, we returned to the sites, monitoring the changes in those 5 seagrass metrics, literally watching grass grow. Finally, to answer our third question, we conducted a short growth rate experiment in which we clipped the seagrass down to the level of the sediment in 3 small quadrats per patch, and then recorded new growth over time. After this, it was time for my favorite part about doing science, the stats. And for the purposes of today's talk, I'm only going to get into the main models we ran for each of the responses for the 3 questions: the initial static seagrass metrics, the changes in these metrics over time, and the growth rates of individual plants. And each of these was a relatively basic generalized linear mixed effects model, where we examined the effect of sea cucumber density on each seagrass response after controlling for things like the distance from patch, sand depth, patch size, and of course, those attention-hogging fish. And since the only p-value I'm interested in is the one coming out of the backside of a sea cucumber, we decided to run our analyses in a Bayesian framework. Now, don't worry if you've never dealt with Bayesian analyses before. The ones we're going to talk about today are virtually identical to their frequentist equivalent, except in how we talk about uncertainty around our estimates. Now, to make a long story short, we couldn't detect an effect of sea cucumbers in either of the first 2 parts of our experiment. For a more nuanced and thoughtful discussion of these null results and their implications, I'll direct you to our upcoming paper. But frankly, I'm not all that surprised we didn't see anything in these parts. Our sampling methods for the first 2 questions, including things like taking cores and cutting shoots, were necessarily destructive, meaning we couldn't track the exact same spot in the patch over time. And we were seeing a surprising amount of within-patch variability in seagrass, meaning that for us to be able to detect an effect at the patch level, the effect sizes would have to be huge. But by following specific shoots at each patch, like we did in our growth rate experiment, we eliminate a lot of that extra variation. So for the rest of our time together, we're going to focus on the model outputs for part 3, because this is where things get really interesting. Now, the plot you're about to see is really just a souped up coefficient plot. So the estimate and the 95% credible interval will be shown as dot and whiskers, but you're also going to see the posterior distribution laid on top, which is just a density plot of all the model estimates for each iteration. As we might expect, growth rates steeply declined at greater distances from the patch, and larger patches tended to have greater overall growth rates. Below ground biomass, corrected for shoot density, appeared to have absolutely no effect on new growth, which provides support for the idea that growth rates are reflective of current environmental conditions rather than past productivity. Fish pee appeared to have a positive effect on seagrass growth, as we'd expect based on past work on tropical fishes. And finally, that's right, folks, we got cool results. Now, coefficient plots with many strong effects like this can be difficult to interpret in a biological sense, so we're going to walk through what this positive result really means. First, we'll pull up the raw data from this experiment. On the x-axis here, we have the sea cucumber density at each of the patches in individuals per meter squared, and on the y-axis, we have the leaf extension rate, or the new seagrass growth per shoot in centimeters. Keep in mind that in our model, we used the log transformed densities, but we're just going to use the untransformed densities here so our results are easier to understand. Now, let's take a look at our model fit for cucumbers at the mean value of all the other predictors. And since we ran this in a Bayesian framework, instead of plotting a confidence interval around the fit, we can plot a sample of the posterior draws, with each line here representing the fit estimated for a single iteration of the model. The positive trend we saw in the coefficient plot is quite clear here, and now we can see how the log transformation of cuc density affects the shape of this relationship. This plateauing reflects our understanding of the saturation effects of nutrients on seagrass growth. There's only so much nitrogen and phosphorus seagrass can incorporate into their tissues before another factor becomes limiting. But Hannah, you might be thinking, look at the spread of your data. There's no way the model fit could be so tight. And you would be absolutely right if it weren't for the other predictors in our model. In reality, the vast majority of the variation in these data is explained when we looked at the effects of all the predictors together. So for instance, we can color each of these points by the amount of fish pee at the patch, and then look at how our fit changes. At any given density of sea cucumbers, patches with lots of fish, shown in lighter shades of blue, tend to have higher growth rates than those with fewer fish. And we can do the same thing with distance each quadrante is from the reef. So shoots that were found closer to the reef, shown in dark green, tended to grow faster than those further away. And so if we step back for a second, I think there are a few key takeaways here. First and foremost, removing sea cucumbers from these patches did not cause a swift and complete collapse of the seagrass ecosystem, based on what we saw in the first two parts of the experiment. But that being said, we were still able to detect an effect of cucumbers on seagrass growth rates, and it was a pretty substantial one. Across the range of sea cucumber densities we saw on Rock Sound, seagrass growth rates varied by more than 20% from the highest to the lowest. And finally, despite existing on the patches in much lower densities than fish, cukes appeared to be equally important in influencing seagrass productivity. This could be because they spend more time hanging out in the seagrass while the fish hang out over the corals, or because by reducing the organic matter load in the sediment through bioturbation, they allow for greater pore water nutrient flow down to the seagrass rhizomes. Whatever the reason, the effect is there. And now, this.
And now, John Oliver perfectly captures Hannah's feelings about the fish that get all of the attention in nutrient research.
For a start, why would anyone be ashamed to be called anti-fish? Fish are stupid. And how do I know that? Look at them. Just look at this idiot. And it's not just him. Check this moron out as well. And while you're at it, what about this dimwit? Here's another bonehead for you. I'm pretty sure this dunce didn't make it past the 3rd grade. And I think we can all agree that this doofus isn't exactly curing cancer.
That's my talk. A huge thank you to everyone who's been involved in this project, and a big thank you to you all for joining me. Goodbye.